I think you are all aware about me and myself. Do you know my name? Okay, my name is Shwandi Bhattacharjee. I basically belong from Jamshedpur, Jharkhand. I've got total of around 8 years of teaching experience, and previous to this, an SMC, Bolpur. I was there as a faculty of Manipal. That's it. I will be acting as an academic coordinator for you guys also. As you can see, it's digestion and absorption of carbohydrates. That's a very familiar topic. You have gone through, you know a lot of things. There are certain additives that we are going to know today in terms of medical perspective. Okay. Starting with digestion and absorption. Absorption we go to the later part, later time. First we'll talk about the digestion. Yes, madam, what you had? What are you having? Yeah, apple. Yeah, okay. Anyone? So what was the carbohydrate component that you had? What do you think? Puri and aloo? Must be. Okay. So aloo and puri. Puri is made up of? Atta, wheat. Fine. Okay. Wheat is coming from animal source? Plant source. Aloo is an animal source? Plant source. Okay. So if it is a plant source, what is the chief component of aloo and wheat or puri? Starch. Very nice. So your major component of your carbohydrate comes as starch. Is that clear? Yes or no? Okay, fine. Imagine if you are going to have any animal product. Not in breakfast, of course. You can. If you are having an animal product, what is the chief component of carbohydrate that you are supposed to take inside? Stored form of carbohydrate in animal is? Very good. Glycogen. Okay. Am I writing visible? Okay, fine. Starch is made up of glucose. Are they polysaccharide or disaccharides? Polysaccharides. Okay. Starch is made up of two components, amylose and amylopectin. Yes or no? Okay. All of you eat rice, boiled rice. How do we boil that rice, if anyone knows? We pour the rice into water, we allow the water to get boiled. After that, we pour that water out. And what we get is a pumped rice that we eat, isn't it? What is that water that we throw? Starch is made up of two components, amylose, amylopectin. There is two components are there. One is the pumped rice that you eat, the well cooked rice that you eat, okay, and the water that we throw. In Bengali we call it as pan or something, yeah. Maad. Hindi mein maad bolte hain. Okay. What is that? What we are eating, which component? Starch is made up of two components, amylose and amylopectin. What is the component that we are having and what is the component that we throw out? What we throw out, the watery part, is nothing but amylose. What we are having is amylopectin. Okay. What is the difference between amylose and amylopectin? Branching point. Okay. Heavily branched is amylopectin. No branching point is what? Amylose. But ultimately the entire polysaccharide is made up of glucose subunits. Okay. What about glycogen? Does glycogen is a branched polymer or a linear polymer? This is very basic thing I'm asking. Please be interactive. Yes, apple girl. Sorry to name you apple girl. The girl with apple. Okay. Yes, madam. Glycogen is sit down. Is a branch structure or a linear structure? Yes, it is. It's a branch structure. What is the branching point? Back boys. What is the branching point for glycogen? What is the linkage? 1:2. There are two linkages possible. Either 1:4 or 1:6. But not is to. 1, 2, 4. We write it as either the branching point can be this or this. Which one? 1 to 6 is what the branching point of glycogen as well as amylopectin. Okay. Anyways, This chemistry part has been already taught to you, so I can expect that you people should know this. The first process of digestion possibly starts from mouth. ठीक है? Am I right? Is my statement correct? Yes, पक्का, पक्का. अरे हाँ या ना बोलो यार. पक्का. सोच के बोलो. What I'm asking is, from next time onwards, I don't want you people to come this late. Make it very sure. Are you पक्का? Sure? 110%? लॉक किया जाए? अफसोस, अब गलत है. पूछो कैसे? आप कोई भी खाना, let it be a plant source, let that be an animal source, कच्चा खाते हो क्या? कच्चा खाते हो? एप्पल को छोड़ दो, फ्रूट्स को छोड़ दो, कोई भी स्टफ, रोटी, गेहूं मिला, कच्चा खा गए क्या? हाँ सर, पेट में जाके तो रोटी बन ही जाएगा. पॉसिबल है क्या? नहीं ना. What is the first process of digestion then? What is the first process of making things digestible? Cooking. Is that clear? It is cooking. Fine. So first process where that occurs outside of our body, which makes any of the foodstuffs digestible, is cooking. Okay? But are we concerned about that? No, we are not concerned about that. How things are cooked or not cooked or well cooked or not well cooked, whatever it may be. Our concern is that how carbohydrates get digested in our system. And the digestion starts from mouth. What our mouth has got? Enzyme. Okay. Which enzyme? Amylase. Amylase? And we specifically, if the amylase is getting released out of the salivary glands, we call it as salivary amylase. You are wrong. You are wrong. Why? Because you have not added a specific point to before that enzyme. That point is adding a name called as alpha or a symbol called as alpha. Remember this thing, okay? Whenever they ask you what is the enzyme that specifically acts on the carbohydrates in mouth, we call it as salivary alpha-amylase. Why we need this specific alpha? Why we need this naming alpha? Anyone? Anyone, please? এই বাচ্চাদের যতগুলো লেটে এসেছে সবাইদেরগুলো নামগুলো নোট করে রাখবে, ঠিক আছে? বুঝতে পেরেছিস? হ্যাঁ, চলো. Yes? What is alpha glucose? I have heard about D or L glucose. Anyone else? Yes, please. No. This alpha orientation or alpha nomenclature provided before the enzyme makes sure that this enzyme is only going to work for alpha glycosidic linkages. Is that clear? So that's why this nomenclature is very needed before you add amylase. Okay? If I say alpha, that means it is responsible to break alpha 1,4 glycosidic linkages. Yes or no? Yes or no? We have in our diet, we generally used to have cellulose. Do we have cellulose? क्या हम खाते हैं cellulose? हमारे डाइट में होता है? होता है यार. कोई भी प्लांट कंपोनेंट खा रहे अपन, उस पर सेलुलोज है. But does it get digested? No. Why? Because the orientation of glycosidic linkage is in beta form. Is that clear? It is in beta form. And what the enzymes we have is specific for only alpha orientation. Is that clear? Is that clear? And these all enzymes comes under the class of glucosidases. Okay, which specifically comes under the nomenclature IUPAC class of hydrolases. Have you gone through the classification of enzymes? Yes or no? Have you heard this name called as hydrolases? You have heard? What is the easiest example to remember for the class of hydrolases? Easiest example. Hydrolases are the class of enzyme. Yes or no? What is the easiest example to remember the names? All digestive enzymes are or comes under the class of hydrolases. What hydrolases does? It adds water and splits the molecule. Yes or no? And these glucosidases can act as endoglucosidases. That means it can act roughly anywhere over the surface or over the structure of any of the polymer. It can act roughly. If we call it as exoglucosides, that means it will be specifically working at the exterior points. Yes or no? But if I am calling them as endoglucosides, that means it is going to non-specifically work anywhere between the two ends of a polymer, of a polysaccharides. And that orientation has to be specifically alpha-oriented bonds. Yes or no? Is that clear? Okay. If we talk about salivary alpha-amylase, the specific or optimum pH lies from around 6.6 to 6.8. Okay? And the mixing of this enzyme with the food is provided by mastication while we are chewing. Okay? Hence, the process of digestion of the carbohydrates or the bulky carbohydrates provided in the mouth is incomplete or partial. Why? Why? Because we don't provide much of the time, we don't provide that much amount of time for the enzymes to mix up with the food and they can act over it. हम लोग generally कितने देरी chew करते हैं? कितने देर chew करते हो खाना को? आधा घंटा? काऊ बन जाओगे फिर तो. In average 5 seconds. In average. ठीक है? उससे ज़्यादा नहीं. 5 seconds is not the optimum time required for the enzyme to act over the entire carbohydrates or the entire polymers of, what we are going to have. Is not enough. As a result, we call the digestion provided in mouth as incomplete or partial. Remember it. Okay? Next, the food moves to stomach. What do we have in stomach? Acids. Is that clear? All enzymes are protein in nature. Yes or no? They are protein in nature. Yes. All enzymes are protein. Fine. Under this high or lower pH or high acidic environment, these all enzymes are going to be inactive. So in total, we call there is no digestion that occurs for carbohydrates in stomach. Yes or no? But one exception is there. It was seen that some of the sucrose molecules may get hydrolyzed by the gastric HCl. Okay? But what I add? Maybe. Maybe. Okay? Is that clear? So in general, in overall, what we say that in stomach there is no digestion of carbohydrates because of the inactivation of the enzyme provided at a low pH. But maybe it is also seen, some studies are there which suggest that no, sucrose sometimes gets degraded provided by the gastric HCl. Sucrose, only sucrose. Okay? Now, this bolus from the stomach moves to duodenum. What is there in duodenum? Is there any opening? Is there any opening of provided in the duodenum? What is that? Common? What is that opening in the duodenum? Coming from pancreas, something opens up? Pancreatic duct. Okay? What releases? What does pancreas release? Pancreatic juice. That juice includes a lot of enzymes and bicarbonate ion. What is that? bicarbonate ion. Now what does that bicarbonate ion performs? What is the role of that bicarbonate ion? See, from the stomach, the bolus that is moving to the duodenum is highly acidic. Yes or no? Yes. So to neutralize that pH, to bring it back to the alkalinity of that bolus or the environment, the bicarbonate ions are released from the pancreas. Okay? So now the pH is somewhere around 8.4 to 8.8 or something upon addition of bicarbonate. And this is the optimum pH needed for the activity of another enzyme that is called as pancreatic alpha-amylase. Here where the maximum digestion of the bigger polymers are going to start, provided by the enzyme pancreatic alpha-amylase. Yes or no? Is that clear? What we had is starch, and upon the action of salivary alpha-amylase as well as pancreatic alpha-amylase, they are broken down into smaller branched components called as dextrins. In some book they write it as limit dextrin also. Both are fine. Okay? They are small branched structures. Small, but branching is there. Yes or no? Is that clear? This enzyme again acts over the food or the bolus or the polymers or the carbohydrates that we had in our diet. But again here the process is partial. The digestion is partial. Fine? So the complete digestion of carbohydrates is going to occur from the small intestine. From the small intestine. We call it as upper jejunum. Okay? And starting from the upper jejunum to the distal ileum, the absorption rate of any carbohydrates decreases. Did you got my point? मेरे बात समझ में आया ये जो point मैंने बोला? Starting from the upper jejunum, going till the distal ileum, from the upper jejunum going till the distal ileum. Okay? If we talk about the absorption rate declines. That means higher rate of absorption is going to occur at the jejunum, upper jejunum. And as we move to the proximal distal side, the absorption and the digestion decreases, provided for carbohydrates. Remember this point. Is that clear? So the maximum form or the complete digestion of the carbohydrates are going to occur in the smaller intestine or the brush border epithelium of the smaller intestine. Fine? Why? Is there any specific thing? Yes, we have specific thing. We call a set of enzymes are present over there that is called as disaccharidases. They are classes of enzymes. There are many enzymes that work specifically on disaccharides. Is that clear? Where did that disaccharide came from? Upon the action of salivary amylase and pancreatic amylase, dextrins are formed. Upon action of pancreatic amylase over the dextrins, some disaccharides are formed like maltose, lactose, sucrose, okay, isomaltose, many more. Some oligosaccharides are also formed, trehalose and all sort of thing. Okay? While they reach to the smaller intestine or the brush border epithelium of the smaller intestine, we have some enzymes called as disaccharidases that are specific for their substrate like maltase, isomaltase, sucrase. They are specific for their specific substrate. And one more, lactase. Fine. So what should be the substrate for isomaltase? Substrate. This is an enzyme. Upon whom it is going to act? Isomaltose. Fine. Isomaltose has got a, what is the structure of isomaltose? Is it branched? Yes. So isomaltase is responsible for breaking the branching points provided what we saw in starch or glycogen. Is that clear, or yes or no? Is that clear, yes or no? Fine. So the enzyme responsible for breaking down the branching point is the complete responsibility of isomaltase enzyme. Fine. Now what we got? We got a series of disaccharides like maltose, sucrose, lactose. These all are specifically broken down by maltase. There is an enzyme maltase that breaks down the maltose. Sucrase is an enzyme that breaks down the sucrose. Lactase is an enzyme that breaks down the lactose. One point I just want to add on, provided to you, that is, as we have talked about endoglucosidases, I have written over there endoglucosidases, right? These all endoglucosidases are generally alpha oriented. These enzymes act on alpha-1,4 linkages. Okay, they are not beta oriented. But lactase is an enzyme that acts beta oriented. But that is not specific for glucose. That is called as beta galactosidases. Is that clear? Is that clear? What I told you, like as we cannot digest cellulose because it has got beta orientation of glucose, but here the beta orientation is of galactose in lactose. Okay, so that's why lactase is an enzyme that has got beta orientation activity provided for galactose. What are the monomers of lactose? Galactose and glucose. And what is the bond? Beta-1,4. Is that clear? So here the enzyme that we are talking about, lactase, is called as a beta galactosidase. Okay, remember that. It is not glucosidase, it is beta galactosidases. Fine? Fine? So upon the action of these all disaccharides, what is the product you got? We started from starch, we got dextrine, we got oligosaccharides and some of the disaccharides. Upon action of disaccharidases, what you got? What is the final product you got? Monosaccharides. What are the monosaccharides? Starch is made up of glucose. You are eating some plant stuff that may have galactose. We have lactose that has got galactose. So ultimately, if you are again eating, she was eating apple, she might be having sucrose. Sucrose is made up of, yes, fine. So you are getting fructose. Some plant foods or fruits generally contains fructose. So ultimately you have got a lot of monosaccharides provided in the jejunum. Now comes the important part of this topic, that is absorption. Remember one thing, if we talk about absorption of carbohydrates, it is only monosaccharides that gets absorbed. Is my statement correct? Because you are studying biochemistry, and biochemistry comes up with, if there is chemistry, what is the next thing we add on? Exception. Yes. So biochemistry, what will be there? Exception has to be there. Yeah, come on. It has to be there. In general terminology, what we say is monosaccharides are the only absorbable carbohydrates provided to our body. But some exceptions are there. It is some of the disaccharides, they in general get directly absorbed from the jejunum through passive diffusion, and inside the enterocytes, inside the cell where they are getting absorbed, they are broken down at a later moment of time. But very rare. In other way, if any of the disaccharides have been provided paternally bole to intravenously or something, anyway, directly consuming or anything, okay, those disaccharides remain undigested and they are getting removed out of or thrown out of your body. They remain undigested. Is that clear? So basically the module or the mode for absorption of carbohydrates is monosaccharide. Is that clear? Okay. Any doubt till here provided for digestion? No doubts? These are all my slides. Anything you just go around, you just go through and let me know if I haven't discussed or haven't taught you about anything that you are seeing over here. Please go through, be fast. Shall I change? Why this is not moving? Oh. Did I discuss? अरे यार इंटरैक्टिव बनो ना थोड़ा, क्या प्रॉब्लम है? हाँ या ना तो बोलो. This slide will be provided to you, don't worry. Yes or no? Anything I left out? What causes the pancreas to release bicarbonate and pancreatic juice provided into the duodenum? Anyone? This way. Physiology, general physiology. There are two hormones. No one, seriously. Secretin and cholecystokinin. Heard about that? No. सुने हो, लेकिन पता नहीं क्या है. Two hormones, specifically secretin and cholecystokinin, that are responsible, that acts, generally cholecystokinin that acts over, the secretin that acts over the pancreas and causes the pumping of pancreas. Okay, that allows the release of pancreatic juice and other components provided into the duodenum. Okay. See, cellulose, as I told, is it digestible? No. So what is the importance of cellulose in your diet? It causes the bulkiness of the stool. It causes proper motility of your excreta. Okay? It relieves constipation. Very nice. Is that clear? If you have seen any of, you must be having elder grandparents or parents, whatever. You must be seeing they have, the doctors generally prescribe them a solution called as lactulose or kind of solutions, okay? That contains cellulose. That contains cellulose. Just to improve the motility, okay? Yeah. There is something written in the red part at the end, absorption rate. Galactose is higher, glucose next to that, and at the least is fructose. I'll let you know why. Okay, we are moving towards absorption now. Yeah. Okay. Point comes as absorption. The major monosaccharide component that you have got over the digestion, what was that? Glucose? Yes or no? It was glucose. So first we are going to talk about the absorption of glucose. Next, we may talk about other transporters and other absorptions or requirement for the absorptions. Let us imagine this is the enterocyte, this is the brush border epithelium, okay? This cell is enterocyte, okay? There is a capillary, a blood vessel, portal vein, anything. This is a lumen. This entire part is lumen, okay? What you had? What you had? Apple, okay? What you had? Apple. That got digested and finally converted into glucose, galactose, let us say fructose. Okay? The first important part that is going to come now regarding the absorption of carbohydrate is absorption of glucose and galactose provided by a specific carrier protein called as SGLT1. Full form is sodium dependent glucose transporter 1. Whenever we add 1, 2, 3, 4, anything to any of the enzymes or protein, that means they have isoforms. They have isoforms. ठीक है? SGLT1 sodium dependent glucose transporter 1 is present in enterocyte, whereas the another isoform that is called as SGLT2 is present in renal tubules. I'll talk about the significance of SGLT2 at the later end. Okay? This carrier mediated transport is active transport. What we need then is active transport. What we need then? When we call anything as active transport, we need requirement of energy. Okay? Now you see, the concentration of glucose and galactose is high over there in the lumen or the enterocyte. Lumen, of course. Okay? Glucose moves in, glucose or galactose, whatever moves in. Okay? It moves with 2 sodium molecules. How many? So it is going to be a cotransport, yes or no? Is going to be a symport, yes or no? Now the point comes, it is generally asked that who binds the transporter first, whether it is glucose or sodium. Anyone? Guesses? Guess number 1, this side. Sodium, glucose, who binds first to the transporter? अरे खाবার দাবার খাও নি নাকি তোমরা? What? Sodium. Okay. This together. Do. No. False. Out of the theory. Okay. This way. एक तो बचा हुआ है, वही बोल दो. ग्लूकोज. हाँ. तुम लोग कुछ बोलोगे? क्या कौन सा बाइंड करता है? सर पता नहीं, कुछ भी लगा दो सर, चलेगा. एब्जॉर्ब होने से मतलब है सर. Any guesses? Sodium. The answer lies as it is sodium. Okay? The transporter appears to be like this. Okay? First of all, 2 sodium binds. Okay? Upon the binding of sodium, the orientation of the binding site of glucose changes. The conformation of protein changes. Upon the conformation of protein, that changes, brings or allows the glucose to bind over here, or galactose to bind over here. Is that clear? Is that clear to all of you? So first of all, sodium binds, that allows or opens the conformation, opens the channel, opens the binding site for glucose, and ultimately glucose and galactose binds. Upon binding, the site opens up at the anterior side and the glucose or galactose is internalized. Okay, with 2 sodium. ये तो facilitated diffusion है. It looks like facilitated diffusion. Concentration gradient के साथ हो रहा है. There we need some facilitator. Sodium is the facilitator. It is opening the gate for glucose or galactose. Yes or no? But I called it as active transport. कहाँ एनर्जी लगा? एनर्जी लगा है कहीं? ATP कहीं दिख रहा है? पर हम तो बोल रहे हैं, I am calling it as an active transport. मैं तो कह ही रहा हूँ. Where is active then? Why we are calling it as active transport? You are getting confused because I have given you a half terminology. The complete terminology is secondary active transport. So the transport of glucose galactose provided by SGLT1 is secondary active transport. When we call any transporter secondary active transport, when the transport of the molecules doesn't require energy, but whatever has come with the molecule, to remove that molecule out of the cell we require energy. And what has come here? Sodium. Sodium should be inside the cell more or outside the cell more? Extracellular. What is inside? Potassium. ठीक है? अब तुमने एप्पल खाया है, एप्पल में 1 मॉलिक्यूल ऑफ ग्लूकोज था क्या? हजारों होंगे, लाखों होंगे, शायद होंगे ना. तो लाखों सोडियम भी अंदर जा रहा है, सेल के अंदर जा रहा है. यस और नो? And as the theory states, the sodium has to be expelled out. For expelling the sodium, what we need? We need a transporter that is called as sodium potassium ATPase pump. Have you heard about this? Fine, yes or no? This pump is responsible for removal of 3 sodium ions out of the cell by internalization of 2 potassium. And for this process, we require breakdown of an ATP to provide energy. Secondary active transport. Yes or no? Yes or no? Is that clear? If we talk about galactose, same module is followed. Same module of transport is followed. Do you remember? I have marked something on red. I told you, like, I will note it down. I'll talk about it later. Absorption rate. Galactose was high. Glucose, then fructose. Bola tha? With this mode of transportation, it was observed that it is not glucose, it is galactose that is readily absorbed by our tissues, by the enterocytes. If we talk about the higher efficiency for this transporter, the answer should be galactose. Multiple choice questions में आते हैं. ठीक है? Next to galactose, if anything that is readily getting absorbed after galactose is glucose, and at the least is fructose. Okay? Fructose is there. Fructose will get inside, provided into the enterocyte with the help of a transporter that is called as GLUT5. That is only responsible for transportation of fructose inside the enterocyte. Okay? Remember this. If we talk about the absorption of pentoses, 5 carbon ring, okay, pentoses, the mode of transport is passive. So it is P4P. Is that clear? The mode of transport is passive diffusion, concentration gradient. If we talk about fructose, it stands for F. The transport mode is facilitated diffusion. So F for F, remember it. F stands for F, fructose facilitated diffusion. Okay. Now the scenario is over here. The glucose, galactose, fructose, whatever they are inside the cell. Okay. Now how do they move outside or outside of the cell into the bloodstream? Where is that? Where is that? Oh. Yeah. This is what I was about to discuss. And it states about some transporters. They are specific for the transportation of carbohydrates. They are called as GLUT. Fine? Glucose transporters. In today's day, in today's era, because of the advanced research and all, we have come across 14 isoforms of GLUT. Earlier it was like, as you can see, we were knowing till 7. But this literature or today's literature of research says that there are at least 14. That much naming is not needed for you. What you need is enough. What is provided over here. Okay? GLUT1, that is predominantly present in the RBC, also predominantly present in the blood-brain barrier, to some extent is present in the kidney. Responsible for glucose uptake in colon, retina, most of the cells of plasma and all. Placenta. There are different forms. You must remember the functions and their, the name of the transporter and their localization. Localization बोले तो कहाँ पे वो present है. It is GLUT2 that is present at the serosal surface of intestinal cell. This glucose that has come into the enterocyte now has to move to the blood vessel with the help of a transporter called as GLUT2 that takes the glucose and galactose into the bloodstream. Yes or no? Yes or no? Okay? So predominantly GLUT2 is present in the serosal surface of enterocytes, lining or that is present adjacent to the blood capillaries. Possibly it is responsible for internalization of glucose into the blood capillaries, or they make into the flow of blood. Okay. Apart from that, GLUT2 has got a very beautiful significance. I am going to make some fun now. Okay. Serosal surface of intestinal cell, they are present in liver, they are present in beta cells of pancreas. Point number 1 I am going to add about glucose transporter 2. They have got lower affinity. No, they have got higher affinity. Sorry. That means, what does it mean if I say about affinity? Have you heard about a term called as Michaelis constant, that is Km? Enzyme kinetics, Km. We write it as Km, short. A general formula is Km is inversely proportional to the affinity of the protein or the enzyme for its substrate. What is Km? It is the concentration of substrate when the velocity of the reaction is at half Vmax. Right? Remember? So, if I say that anything like for GLUT2 has got high affinity for glucose, then what does it mean? High affinity for any transporter, forget about GLUT2. If any transporter has got higher affinity for glucose, what does it mean? Even the concentration of glucose is this much, it will allow the transport. Higher affinity. Affinity का मतलब simple भाषा में प्रेम है. It's love. Love of the protein for its counterpart. Simple. Fine. Is that clear? So if any time I say that this particular protein, this particular transporter has got higher affinity, higher love, that means even this much amount of glucose concentration, if it is present in the blood, it will allow the import or export of it. Okay? In general way, GLUT2 has got a lower affinity for glucose. What does it mean now? The GLUT2 that is present in liver cells or the beta cells of pancreas has got lower affinity. What does it mean? Prem kam hai. The love is less. If the love is less, if you want to show that yes, the transporter is showing to the substrate, yes, I love you a lot. Make sure you are supposed, your concentration should be more or less. More. Fine. Just imagine. Many of you must be liking dairy milk chocolate. You like dairy milk chocolate, right? Okay, fine. I am not going to give you dairy milk. I will give you one Kit Kat. You don't like Kit Kat. Suppose you don't like Kit Kat. How many Kit Kat do I need to give you so that you will get impressed? Aha, sir has given me. Wow, nice. How many? Hundreds? Maybe. De nahi raha hu, imagine karo. Okay. Now if I have to give him a dairy milk, that the chocolate he likes, how many is enough for me to impress him? One. Concept is same. The concept is same. If glucose concentration is high, affinity is low, then only it will go. If glucose concentration is less, affinity is high, it won't allow. Is that clear? Next point. There is something very importantly written over there that GLUT2 is present in beta cells of pancreas. It acts as a glucose sensor in beta cells. अब एक तो फन की बात सुनो. Suppose करो मैंने हाथी जैसा खा लिया है. कितना? हाथी जैसा, एलिफेंट, इतना खाया है, इतना. अब पैनक्रियास से क्या रिलीज होता है? इंसुलिन एंड ग्लूकागॉन. कम टू इंसुलिन. ओके. आई हैव ईटन फूड लाइक एलिफेंट, इतना पूरा, है ना? और मेरा बॉडी और माई पैनक्रियास हैज रिलीज चीटी जितना इंसुलिन. What will happen? Cheaty, you know, and I don't know whether insulin is there or not. Just imagine कि my pancreas has released insulin reciprocal to the size of a cheaty. Will it metabolize my carbohydrates? So my blood will be full of glucose, flooded with glucose. बाढ़ आ गया glucose का. It will start coming out from the urine. Is the metabolism possible? No. Vice versa. I have had, suppose I had चीटी लाइक खाना. चीटी कितना खाता है? थोड़ा सा छोटा छोटा. I had चीटी लाइक खाना. My pancreas released insulin like elephant. What will happen? टाइम नहीं लगेगा. टाटा बाय बाय. हाइपोग्लाइसीमिया गॉन. कोमा. तरंग खत्म. How this is controlled? She had an apple. Apple was having this much gram of carbohydrate. So my pancreas should release insulin approximately or significantly enough to digest that much amount of, or to allow the absorption of that much amount of glucose. Who senses it? GLUT2. The first sensor is GLUT2. GLUT2 has got lower affinity for glucose. ठीक है? So मैंने जब अगर GLUT2 का एफिनिटी अगर बहुत ज्यादा होता है, ठीक है, मैंने चीटी सा खाया है, फिर भी इतना इंसुलिन रिलीज हो गया, टाटा बाय बाय. So GLUT2 has got a lower affinity that states that it will allow the import of glucose into the pancreas only when our blood glucose level is high. Is that clear? Is that clear? Yes or no? ठीक है? And for the uptake of glucose provided into the beta cells of pancreas, ठीक है, these transporters are stimulated. Generally these transporters are present inside the cell. Okay, if this is a beta cell, these transporters are present inside the vesicles. Okay, upon some signal transduction provided by the attachment of insulin, it activates a protein called as AKT, also called as protein kinase B, that is responsible, that triggers the exocytosis of the vesicle, and as a result the glucose transporters comes into the surface and the glucose is internalized. This is how it works. याद रहेगा ये concept? So GLUT2 is a very important component that provides, it is present in liver cells, it has got lower affinity, it is also present in beta cells of pancreas, responsible for sensing the glucose concentration. Okay, fine. Next, GLUT3 that is present in neurons, also in brains. It is primarily responsible, GLUT3 is primarily responsible for the transport of glucose. It cannot transport any other thing. It is responsible only, it has got a responsibility to transport glucose. Okay. Next is another important, GLUT4. GLUT4 is specifically present in, I have missed one point over there. It is specifically present in 3 tissues: skeletal muscle, adipose tissues, heart, cardiac muscles. And GLUT4 is insulin mediated. You got my point? This is how insulin is released. What will cause the release of insulin? Internalization of glucose. That glucose, GLUT2 will sense the glucose. It will allow the release of that much amount of insulin. Insulin will go and bind to certain peripheral tissues. It will cause the activation of AKT. Activation of AKT will cause the externalization of GLUT4 receptors. As a result of that, the glucose molecules are going to come inside the specific tissues like adipose tissue or skeletal muscle or cardiac muscles. Is that clear? So whenever we talk about insulin mediated glucose transporters, be specific about GLUT4. Yes or no? GLUT5 responsible, present in small intestine. It is also present in testis, needed for sperm, also present in kidney. Functionally, it is a fructose transporter and poor ability to transport glucose. Informatively, the presence of GLUT5 plays a very important role in, or the presence of GLUT5 in testes plays a very important role in formation of a functional sperm, because sperm utilizes glucose or fructose? Fructose. Okay? The last one that I've already discussed. Any doubts? Clear? Any doubts? Fine. Some important clinical conditions, like why do we use ORS? When a patient is undergoing diarrhea and all sort of thing, we use ORS. Why? While there is concurrent diarrhea is going on, what happens? We are losing salts. Huh? Our body is losing salt. To replenish that, what we need? We need to give the salt. If salt is given, it won't get absorbed. For that, we add glucose to the ORS solution. Remember this? This will allow the replenishment of the salt as well as glucose that has got drained out from the body, provided due to some condition. Next, this is all about transportation. There are certain medical conditions that we will look around. The most commonest is lactase deficiency, very common, in which there is going to be a deficiency or total loss or total absence of an enzyme that is called as lactase. Lactase is a disaccharidases. It is beta galactosidase that is responsible for breakdown of lactase into galactose and glucose. But in that scenario, if there is a deficiency of this enzyme, what will happen? That lactose is not going to get digested. As a result, the disaccharide will move into the gut. The gut flora, the bacteria are going to feed on the lactose. Yes or no? First of all, lactose is specifically osmotically active. As a result, it will bring water near to it. As it is osmolytically active, it will bring water to it. That water causes diarrhea. Okay, is that clear? After that, if some lactose that has been left undigested that goes into the gut flora feeds over it, that bacteria feed over it, and they produces lot of gas, H2, CO2, they produces lot of gas, H2S to some extent. That will cause lot of flatulence. What is flatulence? Passing of gases. This all together will cause a lot of bloating of the belly, bloatiness. Fine. This will cause irritation and will lead to abdominal cramps. This is the justification that what happens. You can see there are some, wait. Gone. There are some symptoms like diarrhea, flatulence, abdominal cramps, distension and all. This is a justification provided for the diarrhea. Why diarrhea? Because lactose is osmolytically active. It causes the water to come across. Okay, it doesn't allow the water to go back into the tissues. As a result, diarrhea occurs. Flatulence because of the feeding of the gut flora, or the gut flora, the bacteria feeds over the lactose, producing lot of intermediates as well as lot of gases, because they undergo fermentation. That's why gases are produced. Okay, they are anaerobic, so they undergo fermentation. Fine. The release of those gases, excessive production of gases causes bloating. Fine. These gases are released in the form of flatulence that causes irritability, and it also responsible for abdominal cramps. Types, two types, inherited. If it is inherited, there is totally silencing of the gene, totally mutation provided to the gene, no lactase. So the occurrence will be at the childhood. Fine. The moment the baby is born, the first thing is done is the baby is provided to the lap of the mother, and the mother feeds him or her, the baby. From that moment only, the baby will start showing these all symptoms. Okay. If inherited, Other is low lactase activity, that can be primary or that can be secondary. If you talk about inherited case of lactase deficiency, the possible way of diagnosing it is to check the presence of lactose in the urine. Okay, remember, possible treatment that we give, if we notice this kind of case, we give the baby a lactose free diet. It is available in the market. Okay. If low lactase activity, primary low lactase activity, or you can call it as primary lactase intolerance, it is generally seen in non-white population in USA, as well as this is very common in South Asian population like us. South Asian population is very common. It is because of low lactase activity. See how this low lactase activity is caused. Have you heard about a term called as lacoperon? I'm not going to lacoperon, okay. I'm just asking, do you know that genes are regulated? Okay. Now imagine you, like me, I have stopped having milk products from a longer period of time. So what do you think my lactase activity will be? High or low? Low. Why? Because of gene regulation. The body understands the demand. So I have stopped eating or depending on the milk product from many more years. Now suddenly I went under some surgery or anything that happened to me that caused me to depend on milk products on a sudden time. Suddenly. What will happen? What will happen? My lactase enzyme concentration is less. I am having more lactose, more milk product. What it will cause? Lactose intolerance. The symptoms of lactose intolerance. That is due to lower activity of lactase enzyme. And why this is happening? Cessation of having milk products from a longer period of time. It is generally very common in, the elder patients. Elder population generally. My father, if you talk about my father is lactose intolerant now because he has stopped having milk from a longer period of time. Okay, he is lactose intolerant. He cannot eat anything like, paneer or any milk items except curd. Say why? Lactobacillus. Fine. What lactobacillus will do? If you are a primary lactase deficiency, you have got primary lactase deficiency, you had milk, you have milk product, you started with lactose intolerance. What is the possible immediate way of taking an action over it? Immediate way, because you need to rush to the hospital, it will take some time in India's traffic. What is the immediate thing that you can do? Provide that patient with a spoon of curd. It will give a temporary relief at least. Why? Because curd contains lactobacillus. Lactobacillus feeds on lactose. So ultimately, whatever lactose that the gut flora is feeding on will be feeded upon by lactobacillus. As a result, there will be relief of the symptom provided due to lactase deficiency. Okay. Secondary lactase, this can be because of many GI conditions that can be tropic or non-tropical, like celiac disease, kwashiorkor deficiency syndromes, colitis or chronic gastroenteritis, even peptic ulcer provided by, if any surgical removal of any of the part of your GI, that may also lead to secondary lactase deficiency. Okay. There are other things like sucrase deficiency, inherited enzyme deficiency, not common, very rare, but it also happens. Disacchariduria, if there is excessive loss of disaccharides. Okay. Monosaccharide malabsorption, generally due to deficiency or abnormality or any defect in the transporters provided in the intestinal enterocytes or the brush border epithelium. Yes or no? Clear? You should remember lactase, that is lactase deficiency, lactase intolerance, that is more important for you guys. And apart from that, this monosaccharide, urea as well as sucrase deficiency is not much asked. What they will ask you more about is lactose intolerance. Is that clear? Last clinical point I want to add about SGLT2. In these days, in modern medicine, there is a medicine called as dapagliflozin. There are many medicines like that. I'm just giving a brand name. Dapagliflozin. Okay. This specific medicine is used to treat diabetes mellitus type 2, uncontrolled diabetes mellitus type 2, where this drug is a reversible competitive inhibitor of SGLT2. That inhibits SGLT2 in the renal tissues. What happens that if it gets inhibited, the glucose that was reabsorbed, this specific transporter is responsible to reabsorb the glucose from the tubule. It doesn't allow the glucose to go out of the urine. In general, the role of this. But when you take this drug, this transporter is inhibited. As a result, the glucose is released from the body, causing lowering of blood glucose level provided in uncontrolled diabetes mellitus patients. Okay, clinical approach. Is that clear, all of you? No doubts? Thank you.